Good afternoon. My name is Nicholas Hanley, and today I'm going to be talking to you guys about glycolysis, gluconeogenesis, and the role of aldolase in gene expression. The link to my presentation will be included down here as soon as I finish with uploading it to the site. Okay, the objectives for today. First, I'd like to be able to provide with an overview of both glycolysis and gluconeogenesis. Next, we'd like to be able to explain the role of aldolase in both glycolysis and gluconeogenesis. And then we'll distinguish between the various isoforms of aldolase and explain their expression patterns. And finally, we'll identify how the expression of aldolase B is regulated. Okay, so first we're going to start talking about glycolysis. And glucose is a pretty important thing in the way we do our cellular respiration and everything. So since it's the main energy source, it's important that we have a means to break it down and use it. So glycolysis occurs in a sequence of reactions that metabolizes one molecule of glucose into two molecules of pyruvate. Now, this process is important. If we didn't do it in multiple steps, most of the energy would be lost. And this way, by doing it in smaller steps, we can conserve most of the energy in the molecule to be utilized by the cell. This process also produces two molecules of ATP and two molecules of NADH. The ATP is the energy carrier, NADH is an electron carrier. When we're done with this process, once we have the pyruvate molecules, there's still plenty of energy left within them, so those can then be sent on to further reactions depending on the conditions and environment and the organisms we're working with. In an anaerobic environment, glycolysis, the pyruvate molecules produced here would then continue into fermentation. That could either be lactic acid fermentation or it could be alcoholic fermentation, depending on what substances we are working with there. If it is an aerobic environment, the pyruvate molecules will continue on through this process and eventually become carbon dioxide as they travel through the citric acid cycle and electron transport chain. Glycolysis only produces 2 ATP. This aerobic process produces a bunch more if we get through that cycle, but that's for a different presentation. With glycolysis, the whole process is made up of 10 steps. Those 10 steps can be broken down into two phases. The first 5 steps are your preparatory phase, and the next 5 steps, steps 6 through 10, are part of your payoff phase. So if we look at the preparatory phase, and each of these 5 reactions has a different enzyme. As you see in the picture, the enzymes are highlighted blue, and it gives you the molecules that are produced at each step. And when in this part, it's the preparatory phase, we are actually utilizing 2 ATP molecules. We're dephosphorylating them, so we're using those up. So this does require a little bit of energy input here. So in the first step, glucose, through the action of hexokinase and the use of a phosphate from ATP, glucose is converted into glucose 6-phosphate. This glucose 6-phosphate is then converted to fructose 6-phosphate through the action of phosphohexose isomerase. If we look at what's going on, we're just kind of rearranging the way things kind of look on here. And then fructose 6-phosphate, again, this step requires the inclusion of ATP or use of ATP to go from fructose 6-phosphate to fructose 1,6-bisphosphate. So we're going to add another phosphate group on here. The enzyme phosphofructokinase 1 or PFK1 is what's used in this step. Once we have fructose 1,6-bisphosphate, then we're going to use aldolase, and we're actually going to cut the glucose in half. We're going to create 2 3 carbon molecule compounds. So we get aldolase causes that to happen, and we produce glycerol aldehyde 3-phosphate and dihydroxyacetone phosphate. So, end result of the preparatory phase, one molecule of glucose is converted into a molecule of glycerol 3 aldehyde 3 phosphate and a molecule of dihydroxyacetone phosphate. Now, again, we said that 2 molecules of ATP are utilized in these steps, so that's why it's preparatory. It does require the input of some energy. The next 6 steps represent the payoff phase of glycolysis. Here, the molecule of dihydroxyacetone phosphate gets isomerized to a second molecule of glyceraldehyde 3 phosphate. So now we have 2 molecules of glyceraldehyde 3 phosphate, and we're going to proceed through the steps. On this side of the occasion, or the reactions in this phase, it's payoff because we're actually producing some energy molecules here. So if we read the names, you can read them here again. The enzymes are in blue, and the molecules are here, and it even gives you a picture as to what's going on. I just want to note that, notice there's a 2 in front of all of these here because we start with 2 molecules of glyceraldehyde 3 phosphate. Both molecules go through the same process here. So in step 6, we actually are able to produce 2 molecules of NADH. Again, that is electron carrier molecules. That conserves some energy for us. And then steps 7 and 10 both produce 2 molecules of ATP. So this is how we get, we use 2 ATP in the preparatory phase. We created 4 in the payoff phase. So the net result here of glycolysis is the production of 2 molecules of ATP, 2 molecules of NADH, and the 2 molecules of pyruvate here. As we look, there were 3 main types of chemical transformations that occurred through this glycolysis process. First, we degraded the carbon skeleton of the glucose molecule to produce or yield the phosphate. Next, we phosphorylated ADP to ATP in several steps. And then we also transferred a hydrogen ion to the NAD positive to get NADH. So all of those worked out in our favor to do what we wanted them to do. We have a little bit of energy, but the pyruvate molecules, again, like we talked about earlier, still have plenty of energy and can either go through fermentation or the citric acid cycle and electron transport chain to produce even more energy. Each of the 9 glycolytic intermediates between glucose and the 2 molecules of pyruvate that are produced are all phosphorylated. The reason we do that, there's multiple reasons, but one of the first reasons is by phosphorylating the compound, it prevents that substance from being able to leave the cell again. The phosphorylated compound cannot readily pass through the cell membrane. Those phosphoryl groups, second function is their essential components in the enzymatic conservation of metabolic energy. So we pass them to ATP molecules, and that's going to help us pass that energy on. And then finally, what they do is they binding energy is going to lower the activation energy. That increases the specificity of the enzymatic reactions. Okay, now, gluconeogenesis. In this process, again, glucose is a very, very vital fuel for our bodies. Several organs, such as the brain, the nervous system, the erythrocytes or red blood cells, testes, the renal medulla, and embryonic tissues, that is their sole source of energy. So between meals, we have to have ways while we're sleeping to, once our glucose stores have been depleted, we have to find a way to make them release to the bloodstream again so it can provide that constant energy source that we need. So gluconeogenesis helps with that process. It's defined as the method of producing glucose from non-carbohydrate precursors. And basically what it's going to do, if we look in the picture, we start with pyruvate in this case, because that's what we've use most often, and we're going to work backwards and go through these steps and produce glucose. Now, if we would look at all these intermediates here, they're very similar to what we just talked about with glycolysis. There's even 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 steps get us here. So very similar to the reverse reactions of glycolysis. We're going to end up with the same thing. What we start with in glycolysis, um, we're going to end with in gluconeogenesis. So they have opposites there, but they're not just reverse reactions. Um, Gluconeogenesis does occur in all animals, plants, fungi, and microorganisms. The reactions themselves are essentially the same in all the tissues and all the species. However, the metabolic context as well as the regulation techniques used on them vary from species to species and even sometimes tissue to tissue. Um, Animals, the most common starting point are substances like lactate, pyruvate, and glycerol, and occasionally amino acids can be used. Um, in mammals, this process occurs mainly in the liver, but it can also occur partially in the renal cortex, as well as the epithelial cells that line the small intestine. Um, in muscle cells, when they're used, without oxygen, and they use up that, and they can build up lactic acid. That lactate from the muscles can then travel to the liver, where it goes through this process and gets converted back to glucose, where it then enters the blood, goes back to the muscle cells, where it can be stored as glycogen. This cycle is called the Cori cycle, but the actual gluconeogenesis portion of that occurs in the liver. Okay. So we look just if we're comparing the two processes, and we kind of mentioned some of this already, but if we look, glycolysis starts with glucose and ends with pyruvate. Gluconeogenesis starts with pyruvate and ends with glucose. So it would seem they're just the reverse reactions. Um, they produce the same things in opposite directions. However, the mechanisms or the pathways they take to get there vary slightly. Um, so so they're not identical pathways in opposite directions, but they do share several key steps. So 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse of the glycolysis reaction. So 7 of them are identical. For example, the one we're going to be talking about here in a second, here where we use, in this step, we're going to use aldolase to go from um, glyceraldehyde 3-phosphate and dihydroxyacid phosphate to fructose 1,6-bisphosphate. That reaction, this is catalyzed by aldolase, and it does the same thing in both directions. So if we have fructose 1,6-bisphosphate, we can convert it to this and vice versa. So there are 3 reactions, however. 7 of the 10 are identical, just reverse, but there are 3 portions here where we do have some differences. The first reaction in glycolysis, where we use hexokinase, if we look over here, there is a different pathway here to go from glucose to glucose 6-phosphate. The third reaction, where we're going when fructose 6-phosphate gets phosphorylated to fructose 1,6-bisphosphate, it occurs by phosphofructokinase PFK1 here. There is a different enzyme utilized here, so we got a little different mechanism there. So the first reaction, the third reaction, and then the 10th reaction here, when we go from phosphoenol pyruvate, or PEP, to pyruvate. It's done simply by pyruvate kinase in glycolysis, but there's multiple steps here to get from pyruvate back to the PEP molecule here, when we're utilizing gluconeogenesis. So similar pathways, but there are some difference in how we get there, and those differences have to do with energy. The free energy available here is such a negative value on steps 1, 3, and 10 in glycolysis that it's just not feasible to go in the reverse reaction the same way. It's just not going to happen on its own in under the conditions inside the cell. So these other steps here allow us to achieve the same purpose, but doing it in an energy favorable method or manner. Okay, so I said we get back to adolase here, and adolase is the enzyme that causes the fourth reaction in glycolysis to occur. Again, what we're doing here is we are taking the fructose 1,6-bisphosphate and converting it to, so we see here fructose 1,6-bisphosphate, and it is reversible. So this is one of the steps that can occur in both pathways. But if we have fructose 1,6-bisphosphate, we split that into two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So these two are formed by the action. Now, if I have these two in gluconeogenesis, I can combine them through the action of adolase and form fructose 1,6-bisphosphate. So adolase provides us with this. It's the one that actually cleaves the molecule in half. And if we look, it cleaves between the 4th and the 3rd carbon on here. So we have both molecules where we need them to be and get what we want out of it. Okay, the isoforms and expression patterns of adolase. Adolase itself has 3 isoforms A, B, and C. The difference in them results from slight isozyme structural differences and the result in different activities for the different substrates that they're using. The regions where they differ, there are 4 isozyme-specific regions because all 3 of them are homotetrameric enzymes, so they have 4 subunits to them, and a majority of the structure between the 3 compounds, or 3 isoforms, are conserved. However, there are 4 regions that are highly variable among the isozymes, and we call them isozyme-specific regions. So if we're looking in the things, there are 4 regions amongst this where that occurs. The isozyme or specific region, or ISR, the first 3 are found on exon 3, and those 3 predominantly are found in patches on the surface of the enzyme. They don't overlap with the active site, so a change in substrate specificity occurs from a distance, or they can cause the C-terminus interactions with active sites due to ISR4. ISR4 is the most variable of all 4 of the ISRs, and we find that at the C-terminal end of the protein. Now, adolase A is mainly found or expressed in muscle and red blood cells. Adolase B is mainly expressed in liver, kidney, and the small intestines. And adolase C is primarily expressed in the brain, but is also found in fetal and cancerous cells as well. In fetal development, mostly adolase A and B are produced, but newborns and adults, B is the only gene that tends to be expressed. Now, regulation of adolase B expression. So, overall, adolase B can be stimulated to be produced by the presence of glucose and insulin, and it can be inhibited by the presence of cyclic AMP or CAMP. However, there are some transcription factors that can play into this. So the first transcription factor is HNF1 or hepatic nuclear factor 1. Then we have hepatic nuclear factor 3. We have CEPB, which stands for CAAT enhanced binding protein, and DBP is there. It stands for albumin D-element binding protein. So Gregory, Kahn, and Picard in 1993 discussed this, and they looked at these differences in them. And then they tried to figure out how these transcription factors help regulate it. And basically what they found is that the Adelase B proximal region was controlled by at least 5 elements. We can kind of see those differences here, these 5 elements. The DE element, where DE stands for distal element, if we delete this, it ended up in an increase in catalytic activity. So binding here in this region has an inhibitory effect on this particular adelase. Now, we also see that here the CEPB and the DBP site here, this has an activating effect. This CAAT box in here with the NFY seems to have a negative impact on the expression, and then HNF1 and F3 tend to have a overlapping binding sites, so they have a unique interaction that we'll get to. Overall, the results of their experiment showed that DBP and HNF1 had strong transactivators or strong transactivators of Adelase B promoter region. The CEPB and variant HNF1 were weak activators, and then HNF3 alone by itself did not modify much activity. However, HNF3 served as a competitive antagonist or inhibitor to the activation of HNF1, and it also non-competitively inhibited the activation of DBP. So there's lots going on here, and all of these factors lead to whether Adelase B is expressed or not expressed within a cell. Okay, so overall our objectives were to provide an overview of both glycolysis and gluconeogenesis. We also wanted to explain the role of Adelase in both glycolysis and gluconeogenesis. We distinguished between the various isoforms of Adelase and explained their expression patterns. And finally, we finished with identifying how the expression of Adelase is regulated. I thank you for your time, and here are the references.